Today's lesson is on meiosis, and the first thing to recognize with meiosis is going to be the purpose of meiosis, and that is going to be for gamete formation. And a gamete formation. A gamete is such thing as an egg, a sperm. For the process of meiosis to take place, we want to make sure that your sperm and egg have half the number of chromosomes in them. You don't want to have the same number of chromosomes in an egg cell as you have in a skin cell, because when fertilization takes place, that egg and sperm find each other, fertilize, then you want to have the same number as that skin cell would have. So you got to cut the number of chromosomes in half. First thing to recognize also is that the egg cells do not make more egg cells. So this is not a cycle process. Sperm cells don't make more sperm. Inside of the ovaries or inside of the testes, there are precursor cells that are going to be turned into eggs and turned into sperm. In an advanced class, you'll talk about the names of these and go through it. We're just going through the process, but know that the first cell we draw is not a sperm. You don't make the sperm or egg until the end of this process. So we're going to go through the diagramming just like we did with mitosis. So the first thing, we start with interphase. And interphase has a nuclear membrane. It has a centriole. You have your chromatin all jumbled up there. I'm going to draw with blue as my paternal contribution and my pink for my maternal. What's going to happen is there's going to be a replication process just like we did in interphase of the cell cycle. So there is my duplication of my centriole. I now have two of them. And I'm also going to duplicate my DNA. So I'm going to show that. Let's do another round of blue and another round of pink. This looks identical to the cell cycle interphase, so I wouldn't be able to differentiate between the two unless I gave you clues, such as this cell is within an ovary or this cell is within a testes versus this cell is within a tissue in your skin. That then tells you that that cell would have been a interphase of mitosis versus if it says ovary versus testes, this is interphase of meiosis. So the next thing that happens, there are two rounds of meiosis. So to differentiate between them, what they do is they identify it with a Roman numeral most often, so Prophase I. We're going to go through two rounds of prophase. After we Prophase, Metaphase, Anaphase, Telophase. We'll do it a second time. So what we have our cell membrane. Our nuclear membrane is going to start disappearing, hence the dotted line there. My centrioles are going to migrate to the poles. Now what's the major difference here is that when the chromatin condenses into replicated chromosomes, the maternal and paternal contribution, they're called homologous chromosomes. So this replicated chromosome that is blue is really a homologous chromosome to the pink replicated chromosome. What that says is that they have the same genes on them, but not necessarily the same variation of that gene. So this is what's called a tetrad. A tetrad, so there's one tetrad that I drew. I'm going to draw a second tetrad. This doesn't happen in prophase of mitosis. These homologous chromosomes in prophase of mitosis don't interact with one another. So tetrad is not a term you often associate or ever hear when discussing the cell cycle and mitosis. So this is a tetrad. A tetrad is composed of two replicated homologous chromosomes. Homologous chromosomes are the same size because they have the same genes on them, but they're different colors because they come from different parents. So the blue one is a representation of the paternal side of your DNA, and then this pink version is the maternal side. So at the point of prophase I, the two replicated chromosomes pair up. So a tetrad is two replicated homologous chromosomes, and what they do is they line up next to each other. During this process, something happens. This is called crossing over. And crossing over is where a little piece of the sister chromatid that is directly next to the sister chromatid of the opposite homolog, what they do is they switch DNA. So what will happen now is the blue will rearrange itself and the pink will go over there. So they do a little swap. The point of this is to add diversity. At the end of meiosis, we're going to have 4 cells that are produced. 4 sperm or 4 eggs, depending on what type of process you're going through right now. If we assume that this is a cell that's in the ovary at this point, we're going to be making eggs. And so if you have 4 possible eggs that are made at the end, each one of these sister chromatids that we see here in the tetrad is going to be separated. That hopefully makes logic sense that each one of these sister chromatids would disperse into a different egg cell. And with that, if crossing over didn't occur, you would only have 2 variations of eggs. And we want more variation within a species to help with evolution and help maintain that variety. So crossing over adds just a little bit of diversity, because if this sister chromatid were to go into an egg cell, it's now slightly different than this one, because you did a little swap. Remember, homologous chromosomes have the same genes but not the same variation. So maybe if this area on the chromosome was a gene for eye color, these 2 locations would be the same variation because they were once a part of a replicated chromosome. The same location on the pink would be eye color, but it may be a different variation. So now you have a sister chromatid that has portions of the paternal chromosome, but there is a small maternal contribution there. So it does add a little bit of variety. So that's what is called a tetrad. And prophase I is very recognizable due to the fact that these tetrads are present. To proceed then into metaphase I, the nuclear membrane, sorry, change the color. The nuclear membrane would be completely disappeared. We have our centrioles at the poles, and what will happen now are the 2 tetrads that I drew in the previous slide are now going to align themselves at the equator. And still in tetrad form. I'm not going to draw the crossing over any longer. It gets a little too tedious. We'll talk about it at the end. The spindle fibers attach. So here's my equator, this invisible line that separates the cell. So metaphase I, tetrads form or line up at the equator. Another thing we have to know about this is only one variation that could form. There's, depending on how many tetrads you have, this only has 2 tetrads, so there's really not too many combinations that it possibly can do. But this is what's known as independent assortment. Independent assortment says that these tetrads can align themselves in any variation at the equator. So I drew the larger replicated chromosome from the father on top and the mother on bottom. But then for the smaller variety, I put the mother's homologous chromosome on top and the paternal contribution on the bottom. I could have reversed that, and this would just be another option. So it is possible that maybe the next time that the female goes through this process, so a female will go through this process, basically, think about it monthly. Sorry, need to match. There's a possibility that this alignment could happen. They can't happen simultaneously, but you got to think of all possibilities. That's what independent assortment says, is that these tetrads can line up in any fashion at the equator. There's no prescribed method. And that also adds to the diversity of the species. You don't want to have a certain thing that has to fit this pattern, because that would then limit how many variations of egg cells you would produce. This is a very limited choice because we only have 2 tetrads, but imagine a human cell. A human cell would have 23 of these. So the chances that the same pattern would occur first month if a female is going through ovulation versus the second month in a cell is very limited, or probably very unlikely. So from here, the tetrads are at the equator. The next phase is Anaphase I. And so what will happen in Anaphase I, Anaphase is really known for just retracting spindle fibers, so I'm going to retract spindle fibers. I'm going to keep it as if this variation were continuing. I know maybe you drew it a different way because I erased it, but let's see if you can adjust yours. So mine is now the 2 replicated chromosomes that were from the father's contribution to your cell would be going to the northern centriole, and then the mother's contribution is going toward the southern centriole. So they've been pulled apart. In Anaphase I, the replicated chromosomes are being separated from one another. The homologous, I guess you can say, the homologous replicated chromosomes are being separated from one another. The tetrad is being pulled apart. So that's what's happening in Anaphase I. So the next thing we go through is somewhat a combination of telophase and cytokinesis. So Telophase I. So Telophase I. Cytokinesis is that cleavage furrow forming. So there's my cytokinesis picture. The cleavage furrow is pinching in on both sides, but Telophase I, my centriole is still at the north and the south of the cell. My nuclear membranes are now going to reappear. Now I have 2 of them. And whatever was going north stays in this top. So that was the large blue replicated chromosome and the small blue replicated chromosome. And then what was down here, and again, in any fashion or order, the large pink and the small pink. For yours, if you kept up with the first variation, you'd have a different combination of replicated chromosomes. That's the benefit of independent assortment. There's just more varieties that you're actually going to make. So here, that's Telophase I. Eventually, these cells will separate. All right? So let's kind of just adjust that. So let's imagine that we're going to transition right into Prophase II. So you'll notice that we kind of jumped over interphase. Notice there's not an Interphase I and Interphase II. The reason being is that interphase really is about growth and it's about replication of DNA. These cells aren't going to grow, they don't need to grow, and they don't need to replicate. They're already in replicated state. The only thing that has to happen is the centrioles. You can't go through a second round with only having 1 centriole per cell. So there is an understanding that the centrioles between Telophase I and Prophase II, so we're in the second round of meiosis. So now everything's going to have a Roman numeral 2 after it. So to get to Prophase II, I don't really adjust too much with my DNA that's inside the chromosomes, but I do have to double my centrioles, and they don't indicate that with an interphase. That might now be lumped into the Prophase II. Sorry, I cut that as 1. Change that to a Prophase II. Where this nucleus in Telophase I somewhat meant that the nucleus was reappearing, now in Prophase II, this dotted line means that it's disappearing. So you just have to recognize what phase we're in, and how the dotted line sometimes means disappearing, but sometimes it means reappearing. In this moment, Prophase II looks very similar to Telophase II at the nucleus level. The big difference here is we have 2 distinct cells. So in Prophase II, we have 2 cells going on. So what we now do is these centrioles are going to have to migrate. So we'll have them migrated by the time we get into Metaphase II. So let's go to Metaphase II. So we still have our 2 cells. By this point, the centrioles have migrated to opposite poles. In this top cell, we have that large paternal replicated chromosome and the smaller paternal chromosome. Let's attach the spindle fibers. And then in the bottom cell, I have the large pink and small pink. And we have our spindle fibers. So here we have replicated chromosomes line up at the equator, which was different than Metaphase I, where Metaphase I tetrads lined up. It's more about the vocabulary you need to know as to what is aligning at the equator.